Hi, everybody. Thank you for joining the second seminar of the new HIAAD seminars named Bridges, which are seminars presented by two speakers that bridge different disciplines in the field of Alzheimer's disease. The seminar today bridges psychiatry with biology, and it is a great pleasure for me to introduce the speakers of the seminar, Panos Alexopoulos and Ioannis Sotiropoulos. A few words about the speakers. Panos Alexopoulos is an associate professor of psychiatry and psychogeriatrics at the University of Patras and an adjunct professor of psychiatry and psychotherapy at the Technical University of Munich. He currently serves as visiting researcher at the Global Brain Health Institute at Trinity College in Dublin, and his research encompasses early diagnosis and markers of Alzheimer's disease, geriatric depression, age-related sensory loss, as well as issues of philosophy and mental health. The second speaker, who is presenting together with Panos Alexopoulos. The second speaker is Ioannis Sotiropoulos, a researcher, C-level and group leader at the Institute of Biosciences and Applications of the National Center for Scientific Research, Demokritos, in Greece. His research focuses on understanding the orchestrating role of environmental risk factors, e.g. chronic stress, on the onset of Alzheimer's disease, with a specific focus on the relationship between Alzheimer's disease and depression, a stress-related disorder. Combining cell, animal, and human-based studies, Dr. Sotiropoulos studies aims to clarify the diverse factors that regulate the cellular role of tau as a key molecule of neuroplasticity and neuropathology, while his recent work focuses on exosomes as both mediators of brain pathology and potent biomarkers. The title of the talk today is Chronic Stress, Depression, and Alzheimer's Disease, the Triangle of Oblivion. So, Yanni and Pano, the stage is yours.
Thank you. I will start with a very short. You can see my screen. Is it right? The full screen. Can you see it?
Yes, this is fine.
Okay. So let's start the second bridge. The second bridge new HIAAD seminar series. You see where Panos is in the University of Patras. So I think the best photo for that new seminar, I think it is a photo from Rio Bridge that connects the two big parts of the old part of Greece. A bit about what is Bridges, what is Gephyres, this new HIAAD seminar. So as you know, in the previous period, we had two different seminars. The basic research seminars where basic researchers from different, parts of Alzheimer's disease were speaking and exchanging opinions, where there was another one just for clinicians that were speaking and discussing different clinical issues of Alzheimer's disease. So in this second period of HIAAD seminars, we thought that we're supposed to bridge and create some more synergies between different members of HIAAD, as well as other friends and collaborators of HIAAD members, between the three main actions, these actions of HIAAD, which is, first of all, the basic and translational research. The second is the clinical practice and research, as you can see in this nice triangle, and as well the patient care and dissemination. So try to bring this together. Today's bridge is, I think it's very nicely shown by one biologist, me, which is one part of the side, let's say the Republicans, and the other is the Democrats, where Panos, which is a psychiatrist and a biologist, and we're bringing through not easy bridge, I have to admit, because we're speaking for a long time different languages, but as we know now, the synergy between the molecular and the clinical, or let's say the biochemical and the clinical footprints of the disease is giving us the real and better care and real hope for treatment, as we know. And in the middle is the HIAAD as the Holy Spirit, where Lavrendia Grigoriadou, a master student that is shared between me and Panos and Professor Cholaki in Thessaloniki, she's a psychologist, indeed bridging the two parts in a very nice project, as you will see later. So I'm sharing this slide from Vaso Macheraki and Kostas Liketsos, where it shows, I think, the new way we see Alzheimer through a holistic approach in order to understanding the Alzheimer's etiopathogenesis. Whereas you can see in the left part of the slide, we have different molecular mechanisms happening in the brain that can regulate the bioavailability of amyloid beta and tau based on genetics and different cellular availabilities, which they all, as you can see here, act on synaptic dysregulation in different regions and networks of the brain before we come to the establishment of the clinical symptoms that affect, as you know, cognition, behavior, and function. And this is a very long process, which we know now that even 20 years before the appearance of the first symptoms, we have the beginning, the initiation of the molecular underlines that damage the brain. And here we have a big window, as we can say, that there's a lot of attenuators as well as accelerators that can, on one hand, stop this detrimental effect of the molecular effect on synaptic dysregulation, or on the other hand, they can accelerate, like it's chronic stress and inflammation or cardiovascular diseases, which we will focus today on the chronic stress. So this is indeed the triangle we think it connects, because we know that depression is a risk factor for Alzheimer's disease, where emerging evidence from many years now shows that the exposure to lifetime stress or to high chronic stress can also precipitate Alzheimer's disease. And I think here Panos has to start with the same questions. Actually, we're going to ask the same questions, and we try to give the clinical perspective and the basic and translational researcher perspective on the same questions, and this is the issue. So thank you, Panos. You want to share your slide? Okay.
Good afternoon to everybody. Just a minute to share my presentation. So I'm really glad to talk today about the triangle of chronic stress, depression, and Alzheimer's disease from the perspective of a clinician, actually from the perspective of an old age psychiatrist. As Ioannis already mentioned, my talk, or both talks today, are structured across questions. My talk is structured across three questions. The first question has to do with the relationship between depression and Alzheimer's disease. I would like to begin with a definition of my terms. When we talk about Alzheimer's disease, we refer to the most common cause of dementia, but Alzheimer's disease has also a pre-dementia phase called mild cognitive impairment, which is characterized by very mild cognitive deficits which do not severely affect the performance of individuals on activities of daily living. As Ioannis already mentioned, the disease has also a a relatively long preclinical phase, which is characterized by the presence of the pathological hallmarks of the disease, such as amyloid accumulation and neurodegenerative changes, and is also characterized by abnormal biomarkers, even though symptoms are not symptoms, cognitive deficits, behavioral symptoms, and difficulties, dysfunctional and low performance on activities of daily living are not present. When we refer to depressive symptoms, we refer to symptoms like depressive mood, anhedonia, loss of interest, pessimism, psychomotor retardation, energy loss, appetite and sleep disturbances, fatigue. But also we refer to concentration and attention disturbances, cognitive complaints, nihilistic ideas, delusions, and suicidality. Particularly, those symptoms written in red are those which characterize, which are more important and more common in older adults with depression. And suicidality in older adults with depressive symptoms is significantly higher compared to individuals of other age groups. So depressive symptoms are relatively common in AD dementia. Their prevalence exceeds 40%, and the situation is not different actually in mild cognitive impairment. According to studies conducted in community, the prevalence of depressive symptoms in individuals with mild cognitive impairment is higher than 25%, while when we move to clinic-based cohorts, the prevalence of depressive symptoms exceeds 40%. Actually, the presence of depressive symptoms in individuals with mild cognitive impairment does have a clinical significance. Why? Because we know that individuals with mild cognitive impairment and depressive symptoms, they do have an approximately 28% higher risk for developing dementia, meaning that the presence of depressive symptoms in individuals with mild cognitive impairment somehow catalyzes the progression of the symptoms to dementia. And there is the presence of depressive symptoms in older adults without mild cognitive impairment has also a clinical significance, because we know that if we were in the position to eliminate old age depression, then the prevalence of dementia would be 4% lower. So it becomes clear that depression, the presence of depressive symptoms in these categories of people somehow is associated with a more rapid cognitive decline. And this points out that there should be a relationship between the depressive symptoms and the pathogenesis of Alzheimer's disease. And indeed, this is the case because we know that the presence of the epsilon-4 allele of the apolipoprotein epsilon is related to depression and to the occurrence of depressive symptoms. And epsilon-4 allele is the most important genetic risk factor for developing Alzheimer's disease and dementia due to Alzheimer's disease. Interestingly, a relatively recent study has shown that individuals, older adults without cognitive decline, without cognitive deficits, but with late life depression, when they are compared to age, sex, and education matched individuals who do not suffer from cognitive decline or from depressive symptoms, are characterized, those with the individuals with depression are characterized by a higher load of beta amyloid in their brain, as assessed with positron emission tomography imaging employing radiotracing for beta amyloid. So beta amyloid, the load of beta amyloid is one of the hallmarks, one of the biomarkers of Alzheimer's disease. And we see here that individuals without cognitive deficits, without dementia, without mild cognitive impairment, who suffer from late life depression, they do have a biomarker, they do have a positive biomarker which characterizes Alzheimer's disease and which is used nowadays for establishing the diagnosis of Alzheimer's disease. So it becomes clear that there is a relationship between the presence of depressive symptoms in older adults and aspects of the pathomechanism of Alzheimer's disease. So depression is related to Alzheimer's disease, not only with terms of co-occurrence of depressive symptoms and cognitive decline, but also within the frames of relationships between depressive symptoms, and core aspects of the pathogenesis of Alzheimer's disease. I am moving now to the second question, which has to do with possible relationships between chronic stress, depression, and cognitive function. A way to approach stress and extreme stress and exposure to adverse event is to detect in the lifespan of individuals exposure to extreme stress, to traumatic events. And another way is also to assess, to capture perceived stress, meaning how each individual understands, approaches, and conceives actually stressful events in his or her life. I am beginning with exposure to extreme stress, such as the Holocaust. And when we compare older Holocaust survivor to older individuals without exposure to war-related traumata, it is important to underscore that both groups did not suffer from cognitive decline, dementia, or mild cognitive impairment. There are significant differences between these two groups with regard to depression symptoms, to post-traumatic stress disorders symptoms, meaning symptoms like re-experiencing the traumatic event, avoidance symptom, hyperarousal, or increased emotional and physical reactivity. And also, the two groups significantly differ with regard to gray matter volume in brain areas like the orbitofrontal cortex, the temporal pole, the prefrontal cortex, and the angular gyrus. So, extreme stress, traumatic experiences, somehow many years after the exposure to the stressful event, are related to differences in with regard to depressive symptoms, to PTSD symptoms, and to brain and to the volume of brain parts. If we consider also cognitive function, then we do have the triangle on which we focus today. We do have the triangle of childhood trauma, meaning extreme stress, exposure to extreme stress. We do have depression, the presence of depressive symptoms, and we do also have a lower performance on a task of delayed face and name recall. And what is really interesting is the fact that lower cognitive performance on this delayed face and name recall task is not related to the reduction to the decrease in hippocampus volume, but is related to the presence of depressive symptoms. So we do have this clear connection between stressful events, adverse events, depressive symptoms, and cognitive deficits. But this is actually the one side of the coin. We do have also another way to approach adverse events or stressful situations, which has to do with the assessment and the capture of perceived stress. Individuals who rate the 10 past years of their life as extremely stressful, they do have a higher risk to function poorly on the mini mental state examination compared to individuals who do not rate the 10 past years of their life as stressful. And this was a study which was conducted in the UK, and the recruitment of the individuals was based on the support of their general practitioners. When we move now to low and middle income countries, and based on the findings of the World Health Organization study on global aging and adult health, there is also a clear relationship between perceived stress and the phenotype of mild and cognitive decline and the phenotype of mild cognitive impairment. Here, perceived stress was assessed with the perceived stress scale, actually with 2 items of this scale. And according to the findings, a 1 unit increase in the perceived stress score resulted in a 1.14 times higher odds for mild cognitive impairment. So stress is related to brain volume changes, is related to depressive symptoms, is related to lower cognitive performance, and it also pertains to the phenotype of mild cognitive impairment. Let's move now to the third question, which I will try to answer. Could stress be an important etiopathogenic precipitant of depression and Alzheimer's disease? I think this is the central question of our seminar today. In order to label an event as a precipitating factor, there should be a temporal sequencing. There should be an event which occurs firstly, and then we do have the occurrence of the other events. In our case, stress is the first event, and then we do have the occurrence of depression in older adults and the onset of dementia symptoms. There should be a clear, statistically, and in my case, also clinically significant relationship between these 3 factors of the triangle, and there should not be other factors, moderators of this relationship between stress, depressive symptoms, and dementia symptoms or symptoms of mild cognitive impairment, which means that we have to justify or to establish a causal relationship between stress, dementia, and stress and the development of depressive symptoms. And indeed, there are clinical data which point to the fact that the higher the number of post-traumatic stress disorder symptoms that are present, the more rapid the cognitive decline of these individuals is. Based on the findings of a community study which included middle-aged female nurses who were cognitively assessed every 6 months, those individuals, those female nurses who suffered from more PTSD symptoms had a more rapid cognitive decline compared to those without PTSD symptoms, or with less PTSD symptoms. And exposure to extreme stress, exposure to traumatic experiences, traumatic events is also related to an increased risk for the development of dementia. And what I find particularly interesting for psychiatrists is the fact that the risk for developing dementia was found to be higher in those who were exposed to traumatic events in the society, in the general population, and not in veterans, meaning that somehow traumatic experiences which are not related to war experiences, to combat fields, to have a more detrimental, a more significant negative impact on progression, on cognitive decline and progression to dementia. Perceived stress is also high scores on perceived stress are also related to the development of mild cognitive impairment. Individuals with high perceived stress, they do have a 30% greater risk for developing mild cognitive impairment compared to individuals with low perceived stress. And this finding is based on a follow-up period of approximately 7 years. Also, perceived stress in midlife seems to be related to dementia risk, according to the findings of a relatively large Danish study, which included more than 10,000 individuals. In these individuals, perceived stress was assessed in midlife, and they were then followed up with regard to the establishment of the diagnosis of dementia based on data of different Danish registers. And thus we can see individuals with high and medium perceived stress had a higher risk for developing dementia compared to individuals with low perceived stress. Perceived stress is also related, pertains also to depression risk in older adults. According to the findings of the Irish longitudinal study on aging, individuals in the highest quartile regarding perceived stress, they do have 2.65 times higher risk for developing depression within a 2-year, within a follow-up of 2 years. So stress is related to high risk for cognitive decline, dementia, mild cognitive impairment, and depression. But in order to justify a precipitating role of stress for the development of depressive symptoms in older adults, and also for the onset of dementia or MCI symptoms, there are open questions which should be approached and which should be answered. For instance, are there relationships of stress levels with biomarkers and their biomarkers of Alzheimer's disease, and their longitudinal changes? Can stress serve as accelerator of biomarker positivity and cognitive decline? Is there actually an influence of a 4th, 5th, or 6th factor on this triangle of stress, depressive symptoms, and onset of dementia or mild cognitive impairment? I think that Ioannis in the next minutes try to give answers to these very central questions for justifying this possible precipitating role of stress for the development of depression and the development of clinical phases of Alzheimer's disease. Many, many thanks for your attention.
So I will continue where exactly Panos left. So we're going to see after the excellent presentation of Panos, which, as we said before, I'm going to give you the basic and translational answers to the same indeed questions, trying to provide evidence that comes to questions from the bench, from the bed to the bench and back to the bench. So is there a relationship between chronic stress, depression, and cognitive decline, which you listen the question, the clinical evidence for that answer from Panos? So what we think now, after 20 years work starting at Max Planck Institute of Psychiatry in Munich, which is basically the center for depression, where I start this part, we think that chronic stress through the same neurobiological mechanisms can connect between depression or mild cognitive impairment and AD by common neurobiological mechanisms, which they trigger and they damage neuronal malfunction and atrophy through the overlay. So it is like stress. It works like, a push down, a weight that is put in this normal aging line that, by time our mood and cognitive status can decline a bit, through really down, through pathological aging, through common biological mechanisms, as we will see in the next slide. So here on the left, you can see in a very simplified way the pathways and what I try to summarize to you. So in order to answer this question, we exposed our animals, our experimental animals of Alzheimer's disease, which is for sure it's a model. So they have increased amyloid beta or tau, hyperphosphorylation, which is this molecular characteristics, histopathological characteristics, and molecular actually characteristics of the disease in their brain, and we expose them to big periods of chronic stress or high levels of the main stress hormones, the glucocorticoids. And to make things simple, we see that chronic stress can indeed activate the molecular mechanism that leads to the overproduction of amyloid beta, this peptide that it is, I told you, a hallmark of the disease. And then we also see that stress can also, and amyloid beta is known to do that, to trigger tau hyperphosphorylation and accumulation, as you can see in the neuron, which is also a very typical characteristic of the AD brain, as well as in the synapses. And different work from us and others has shown that chronic stress and high glucocorticoid levels in animals can hyperphosphorylate tau, which is then missorted, abnormally accumulated in the synapse. And we have here electron microscopy evidence and multiscale levels that indeed give an activation to NMDA, and more specifically glucuron, to be excitotoxic signaling in the spines. That creates a lot of toxicity signaling into the spine that leads to the atrophy and neuronal malfunction and memory loss, as we know now. Where on the other hand, through the same mechanism, by the overproduction of amyloid beta and the hyperphosphorylation and damage of tau protein, which is the cytoskeletal protein in our neurons in the brain, we also damage the other very important mechanism of neuronal plasticity that is important for the memory and the cognitive status of our brain, which is the adult neurogenesis in our brain. I don't want to go to details because this seminar is trying to simplify things as we try to bring people from different backgrounds that are all focusing on AD. So all these molecular damages, as I told you, lead to synaptic malfunction, atrophy, and neuronal malplasticity that leads to cognitive and mood deficits. But very, very importantly, and we can do this in experimental animals, when we remove the protein tau, we were able, different studies from our team shows that if we remove the protein tau, we can block all the synaptic damage and memory and mood deficits that chronic stress induce, suggesting that this mechanism that is known to be found in Alzheimer's disease is also found in stress-driven brain pathology, in stress-driven depression, in other words. For sure, we are also always speaking about experimental studies, okay? With all the limitations that we can use, that we can use experimental animals to mimic depression. So what we usually do, we use prolonged periods of stress, and this is a model for mimicking depressive symptomatology and brain damage found in depression in mice. And this is the evidence that if you don't have tau, I don't want you to go in very complex information, just read the titles. We don't have anymore the damage that stress doing on the synapse, on the communication between the neurons and the memory establishment of that. We don't have the atrophy that stress causes if we don't have tau. We also don't have this MRI damage that we can see by MRI, both in humans and in mice, that stress is doing if we don't have tau protein, and as well, we don't have the memory deficits. Again, suggesting all this information that tau is essential, damage of tau is essential for the damage, the detrimental impact of chronic stress on our brain under depression status, at least when we model it to animals. So now to the next question that also Panos provide you some clinical evidence supporting this answer is, could stress be an important etiopathogenic precipitant of depression and Alzheimer's disease, connecting the two diseases? And again, the answer is yes, because we see that chronic stress, again, when we expose experimental animals, experimental models of AD or depression in chronic periods of stress, long periods of stress, 6 weeks, 8 weeks, this is big for the short life of 2 years of animals like mice and rats, we see that this aggravates the amyloid beta pathology here, and this results to synaptic damage and memory deficits. And even further, we also tested some new drugs. I think now it's in clinical phase 2, this glutamatergic signal AMPA receptor, let's say more specific, drug that can attenuate, again, very close to the clinical conditions where we use memantine for protecting the excitotoxicity that occurs in the AD brain. And memantine, as you know, is used in patients in the later stages of the disease. Furthermore, we also saw, and you can see again in this little cartoon on the left, that even in Alzheimer's disease model, we see that chronic stress and high glucocorticoid levels, the main stress hormones, aggravate different parameters of tau pathology, of human tau pathology, the same one that we find in human brain. We also can mimic it in Alzheimer's disease, such as the accumulation of tau, the neurotoxic aggregation of tau that is very typical for the Alzheimer's disease brain, and the cell death, and as well as other mechanisms that we more often find now in human diseases, not only in Alzheimer's, but also in others that have to do with stress granule formations. And this is a molecular mechanism that helps tau, synuclein, as well as other proteins, TB43, to aggregate in toxic forms inside the neurons, leading to neurodegeneration and finally the cell death. So in other words, I show you different experimental evidence that shows that chronic stress, either in depressive models or in Alzheimer's disease models, can aggravate the whole damage of the brain, triggering the same molecular mechanisms that are found in Alzheimer's disease, also in stress-driven depression or stress-driven brain pathology, at least in experimental studies. Now, the other questions that also Panos referred to is that if sex-dependent responses to stress explain the increase of Alzheimer's risk for females, because we know that women has higher risk to develop Alzheimer's disease, where when they suffer from Alzheimer's disease, they have higher cognitive deficit and bigger damage. So in another experimental studies, we are trying to really understand how different risk factors of Alzheimer's disease can interact in order to precipitate the disease, and more specifically, the pathology of brain pathology related to tau that we found in Alzheimer's disease. So we know that aging is a very important parameter. Risk factors, we need some mutations that we know that also they also have a significant risk factor, as well as sex, the female I told you, and the stress. So what we did, we use a transgenic animal that is a model for tau pathology in mice, the model of the human Alzheimer's disease brain pathology, at least the tau part of it in the mice. And we use both sexes, female and male, of different ages, like middle age and old, really old. You see 2024. This is really like the end of the life for mice. And we try to understand how these interact, because we know, for example, that AIDS animals or AIDS people has a different difficulty to cope to stress, so they are hyper responsive to stress. So in very little, and without making a long story short, we find that if we reduce the amount of stress, we see that female animals were more vulnerable to stress, in line with the clinical evidence supporting that women has a high risk for Alzheimer's disease, and that this has to do with the, let's say, increased vulnerability to molecular toxicity of tau of the hippocampus in females. In other words, we see that when we reduce the amount of stress we exposed our animals, female or male animals, that female were more vulnerable, and this was seen both in memory deficits that were higher in female, as well as in the molecular toxicity and cell death that is higher under stress conditions in female animals. And we also try to find a molecular mechanism, but this is, when you leave from humans to mice, molecular mechanisms can be also different to clarify. But as you see now, we have some evidence based on biological data that they detect amyloid beta and tau in the brain of living humans, where we see that in non-demanded conditions, when we have stress, we have increased amyloid beta or even tau. There's another more recent studies that tau by PET images is shown to be increased in depressed patients in some specific areas. So the last question that I want to share with you today is how stress accumulates pathological forms of tau. And very fast, there are two basic mechanisms that we seem to aggravate this stuff, and has to do with the endolysosomal pathway and the autophagy. So making again a big story short, our studies have shown that chronic stress can damage the mechanisms that degrade tau protein, that degrade protein, and let's say cut and remove tau, bad pathological tau from the neurons. These mechanisms are inhibited, are damaged by stress, and this results to the accumulation of tau, of pathological forms of tau inside the neurons. And this we know that is very toxic, and this is a hallmark of the Alzheimer's disease in human brains, the accumulation of pathological forms of tau inside neurons. However, these mechanisms are also connected to exosomes. So these results leads us to the next question, which is, could chronic stress provide a biomarker footprint for its AD precipitated role? The question that I think Panos was the last part that should be further cleared. And I think we know that very short about exosomes, which seems to be secreted by the diseased neurons and spreading the disease to the next healthy neurons and transforming to humans. And we tried to work with this and try to, if we can get this information. So because there are difficulties working with us, we make a new protocol that advance the way we can isolate biomarkers or potential biomarkers such as exosomes. And in this story, I'm just showing you very briefly the work we have developed together with another colleague and member of the HIAAD, Dimitris Ioannis from Johns Hopkins Medical School and NIH in Baltimore, as well as Bruno Costa Silva. I don't want to stay in details about how we developed this new technique that provide us better exosomes, these potential biomarkers for the disease. But I won't tell you that this is published, so you can go and see it. One, and the second is that it works. It's much better technique and provides better exosomes for both mouse as well as human brain. And I will go back to the question whether this is applicable under stress conditions. And yes, the questions we are giving now is that, you can see in this slide, sorry, there was a mix in the slides, that chronic stress increased brain exosome secretion, leading to memory deficits. We're analyzing in animals again, we're analyzing what these exosomes, these small secreted vesicles that are thrown out from neurons, and they help the propagation of the disease of Alzheimer's disease in the brain, because they carry amyloid beta or tau in the AD brain, but also in the mice, as you can see, what they transfer. So did stress put more of these pathological proteins inside the exosomes in order to transfer and increase the brain pathology in the brain? This is the way, this is the analysis. It's ongoing in the lab with different complex proteomic analysis we're doing together with Martina Samyotaki and Dr. Panayotou at Fleming Institute, as well as some microRNA and RNA-seq analysis of the content of the exosome as well as of the brain, because in order to have a good or let's say to explore the biomarker potential of these exosomes, you need to understand what is the brain pathology, where these exosomes come from, and then see, do they transfer the same molecular signature? Then probably could be good to test it in clinical studies to see if this molecule or this pattern of molecules can be good for as biomarkers. And this is something that I would like to show you, that yes, exosomes seems to be a very promising tool. And here you can see a recent publication, a review publication actually from us and Dimitris Kapoyannis and Bruno Costa, as well as Konstantinos Koulariki and Panos Vlamos, a core also member of the HIAAD, where we see that during the progression of Alzheimer's disease, we see through all the healthy preclinical MCI, which is a prodromal phase, as you heard from Panos, we have up to the Alzheimer's disease, we have an increase of proteins, very similar way to very similar proteins that we have in the AD brain. We also have this increase in the exosomes, which can be collected in the peripheral brain blood. And Dimitris Kapoyannis, and now we're using the same protocol, have a very excellent protocol of how to collect brain-derived exosomes in the peripheral blood. And here, look what different studies have shown, including studies from Dimitri and others, that in these exosomes collected in the peripheral blood coming from the brain, we have an increase of phosphorylated tau, of amyloid beta, very similar of what happens in the Alzheimer's disease brain, inside the brain, as well as reduction, for example, of synaptic proteins, because I told you that in AD, independently if there is or there's no stress, we have a loss of synaptic function, atrophy of synapse. That's why we cannot memorize and we have problems in memory, which you can see here. The same synaptic proteins are also reduced inside the exosomes, suggesting a very great potential for biomarker tool of exosomes, of brain-derived exosomes that we can even collect not only in the brain, but also in the peripheral blood. So I think I'm almost finishing with the real bridger, which is our student, Lavredia Grigoriadou, that he has collected this AD cohort in Thessaloniki the last 2 years, which is basically clinical data and blood and saliva, and blood, plasma and saliva. And here, I don't know if you see my slide, we are trying indeed to answer a bit with this clinical study that has about 200, 220 participants with different, high. We try to answer this question whether if we can understand the fingerprints of anxiety and stress levels in patients with MCI and AD dementia, if we can see any difference in their exosomal. And for that, we're using a good clinical characterization. And we also use anxiety and stress levels, both based on the clinical data, but also measuring cortisol saliva samples from 3 measurements during the day that give us a better overview of the high exposure of this person to the cortisol, to high glucocorticoid levels. And we also do blood analysis with exosome analysis. We are a bit behind on that due to some technical issues, but hopefully with Martina Samionaki we'll find some. And also we do some cell-free DNA polymorphisms from the blood, the cell-free DNA that also exists in the blood, besides the one that exists in the exosomes, trying to understand where we have a different proteomic or DNA signature between all these categories of patients, or these clusters, to be absolutely honest, with the low stress and the high stress, try to see if we can give answers even to the clinical levels. And here I would like to close and just remind to our audience that the HIAAD, and in a very great collaboration with Crete University Press, has released the last 2 years, 2 books about Alzheimer's disease. One, the first one it's called Όταν η λογική κυνηγάει τη μνήμη. It is very simple written. It is for the general audience, and you can read there all the different things you listen today and in other presentations of HIAAD, written very simply in individual chapters of 5 to 7 pages. And you can have more information about all of these issues, as well as practical issues of where to go, where you can help your people. And recently, in 2022, the HIAAD, together with Πανεπιστημιακές Εκδόσεις Κρήτης, also released a second book, which is really gives some practical advice about how you deal with a patient, as well as a caretaker and the family of a patient, which is very important. And this is more focused to people working with or living with patients. And after that, I think I'm done. I would like to thank different people from the joint lab, what we called, here in Institute of Bioscience and Application, as well as my old lab in ICVS Institute in Munich, in Minho, in Portugal, where we work together because we can find together, and all our collaborators, including Panos Alexopoulos, we have started recently collaborate, and a great speaker. Thank you so much.
Thank you, Yanni. Thank you, Pano, for these excellent presentations, bridging chronic stress with the biology of Alzheimer's disease. So the speakers are available to questions that the audience would have. Please.
Do we have any questions on the YouTube too also?
Questions. Raise your hand so that we can see the questions. Yes. Kostas, please. Kostas Liketsos would like to make the first question. Kostas, please.
Hi, everybody. Really nice. I like how this is bridging things. Would someone care to tell me the pathology of depression? Because we're talking as if we know it.
Yeah, the pathology of the pathomechanism of depression in old age is related to deficiency of serotonin or norepinephrine in the synapses, is also related to a reduction of growth factors, is also related to inflammatory processes, as well as to a dysregulation of the hypothalamus pituitary adrenal axis. And there is also field for further research, since we do not really have captured all,
and this is
all the aspects of the pathogenesis of depression in older adults.
If I can add here to the question, and based on these 4 or 5 different parameters that Panos referred and connect to depression, all of these are, let's say, triggered by chronic stress, and we know that both in humans as well as in animals, and that's why chronic stress is used as the model of depression in animals. For sure, it's a model, it's not perfect. And Kostas, you're absolutely right. We don't know what is the real human pathology of depression in the human brain. We have some issues. We have some actually insights or indications, but we can only mimic and hypothesize. However, the common neurobiological mechanism or molecules that can connect depression with AD through chronic stress, it seems, or not through chronic stress, but at least they are common. There was more basic, or let's say experimental evidence, and now I can see more and more as clinical studies and clinical researchers are moving away from just the clinical characterization, but going to the and following to the NIH guidelines to focus more on molecular biochemistry of the brain and the patients. We see more commonalities.
Yeah, I raise this because I find it to be a common mistake. Depression is fundamentally a set of symptoms. And so I don't understand how we believe a set of symptoms can cause anything in the brain. Ultimately, this set of symptoms is the result of one or more interrelated brain processes. And Pano, you framed them very nicely. So I encourage people in this field to move away from studying the links between a set of symptoms and brain pathology and start introducing to the study the underlying brain mechanisms that produce that set of symptoms. So it might be, should it be the inflammatory pathway? Should it be related to age-related loss of core monoamine neurotransmitters? So all of the above makes sense. It's just that it doesn't fit my mind to say that symptoms cause brain pathology. I don't
fully agree.
The logic. Yeah,
we fully agree.
Ioannis Zaganas.
Yes, thank you very much for the nice presentation. So I was wondering, so you very nicely showed, both of you, that stress or depression precipitates Alzheimer's disease. But do you think there is enough evidence that if we treat stress or we treat depression, we can, let's say, reverse the pathology that leads to Alzheimer's disease? I think that's the big question we have here. So either on experimental level or on epidemiological
Both Panos and
level.
Kostas has a lot of experience on that one, sir.
Yeah, well, Ioannis, what I think is that we need more studies, and we need particularly interventional studies, which hopefully will show us that the treatment, the management of, for instance, stress symptoms or PTSD symptoms somehow influence or change, modify the rate of cognitive decline. This would be a step of paramount importance for establishing a causal relationship between stress and PTSD symptoms, depression, and AD, dementia, or cognitive decline related to Alzheimer's disease.
Kostas, do you have a comment on that? Because we have discussed this in the past about the use of antidepressants, if they're beneficial or not.
Yeah, I mean, it takes a while to sort of get into it, but it speaks again to the presumed underlying mechanism. So Panos mentioned loss of monoamines. I think the interesting question would be if depression in a prodromal state is a marker of a monoamine pathology, and the monoamine pathology is interacting with the ATN pathology, then perhaps using a serotonin augmenting drug medication would improve and break the cycle of interaction between loss of serotonin and the ATN pathology. So that would be my general comment. And we're trying to design a trial along those lines. In our trial, we're going beyond depression. We're looking more at this MDI construct, which would include late life onset, somewhat atypical depression, where you don't have the suicidal thoughts and the self-deprecating thoughts, but you've got more of an anhedonic, apathetic, neurovegetative disturbance.
So in this regard, if I may add, maybe it's not beneficial to have zero stress also. So maybe it's the level of stress that you develop that's important. So if somebody doesn't have at all stress, or someone has a high levels of stress, both are, let's say, problematic for the development of Alzheimer's. So I don't know if you can talk about this. So you both nod affirmatively. So we need to let's say, define what's the level of stress that's beneficial for everyone.
That's why they're measuring depressive.
Interesting point. We can move to the next question by Maria Basta and then Rebecca Macha.
Well, congratulations to both presenters. They were really good, and they matched very well together. I was having the same. I was gonna add something about that antidepressant, but Dr. Liketos covered me. Unfortunately, he spoke before me. But it's true that studies up to now have not proved that antidepressants are helpful for memory issues in everybody. So I think there's, as you already mentioned, there's several factors, and it depends on the case where it can help and where it doesn't. Actually, another component of this story is insomnia, which is also related to HPA axis activation and depression. And this is also a factor that is associated with memory decline in older people, and not only in older, but in general. So I am convinced that the HPA axis and the stress and the cortisol and all this do have some role, but it's not the only mechanism that can explain these issues. But I do believe what Panos said, that interventional studies on psychotherapeutic stress management interventions are going to give much more light into the issue that we're talking about. But as far as I know, there isn't much out in the literature yet. So this may give some really good understanding of the interrelation of all these things. So congratulations again.
Thank you, Maria. And again, I would like to highlight the Kostas comment, the previous Kostas comment, that these kind of studies should, and that's why we do this kind of presentations. These clinical studies has to really be supported by biochemical molecular analysis of the improvement, if there is, and at the molecular level too. So I really would like to highlight Kostas' comments on that, based on the fact that we need molecular evidence. And just one comment, and I'm finishing, that yeah, there is very clear studies showing even in healthy populations that antidepressants can reduce amyloid beta levels in humans and in mice, as well as the secreted part of amyloid beta. Now, when we are giving the antidepressants, I'm not the expert to answer, so most Panos, Ioannis, and all the clinicians and Kostas could say about that, but I think a critical time window, and I want to share this with you, is that more and more clinical studies focus in the early phases of the disease, even in the pre-symptomatic phase of the disease, in order to revert synaptic damage and neuronal malfunction early before it is irreversible. Because my view, based on what I'm reading, again, I'm not a clinician, is that when we try to intervene, when we try to give a therapy or a treatment, we are too late. We are going there we cannot reverse the neuronal loss. There's no Lazarus drug so far in the brain, if you know what I mean. So that could be something important. Probably the clinicians could give more feedback on that. Thank you.
Next question by Rebecca Matza, please.
Hello, very nice talks, Yanni and Panagiotis. I think we're very convinced, you were very convincing to show us that depression is sort of precipitates Alzheimer's. My question is this an accelerator? A fact that has something to do with time? Are there converging mechanisms in a way between depression and Alzheimer's? What is your idea that if you have depression, for example, this does something to your brain that is somehow similar to what is happening at the early stages of Alzheimer, or are these two totally different things? Because somehow you can have Alzheimer's without depression as well, I guess, right? So I don't know, this was my question.
Well, Ioannis, may I try to answer the question? Yeah, well, Rebecca, depressive symptoms are relatively common in individuals with Alzheimer's disease, but they are not present in all individuals suffering from Alzheimer's disease. Depressive symptoms are not a presupposition for establishing the diagnosis. It is also my question if there is clinical evidence which points actually that the presence of depressive symptoms in individuals with mild cognitive impairment somehow accentuates, catalyzes the rate of cognitive decline. If the presence of depressive symptoms or the pathogenesis of depression somehow influences the core pathomechanism of Alzheimer's disease, I think that Ioannis could answer the question, but in my opinion, we need more evidence in order to be sure that there is a causal relationship between stress and the onset of depressive symptoms, and there is a causal relationship between depressive symptoms and accentuation or the onset of dementia symptoms. Because there is also the hypothesis that somehow, particularly in early stages of the disease, that anxiety or that depressive symptoms form a precognitive symptom of Alzheimer's disease. So I think that the questions remain open, and we have to work on so that conclusions can be drawn.
I'm not an amyloid beta believer, and I fully agree with Kostas on that. And I think that's why there will be no good future for these new drugs on amyloid beta. But now I want again to challenge Kostas, because that's a nice debate, especially for younger students, to see clinical practice and experience of many years from all the clinicians back to the experimental models, because an experimental model can be an experimental model. So Costa, can you please give us what your opinion about what Panos was saying, that whether the depression comes together with MCI or dementia? Or, in other words, depression is just the symptoms, as you said. Does it mean that we have one unique pathology or whatever damage in the brain in what we call depression? Because there are different types of depression too. One, and the second can be together with Alzheimer's disease, molecular underlying damage on the brain, or can be before?
Yeah, this is opinion, clearly, but I think opinion that can guide research. We have to introduce a time dimension. So, and perhaps also a phenotype dimension. So the time dimension has to do with the first onset of the depressive symptoms. Generally speaking, if the onset of depressive symptoms is well below old age, so old age is anybody older than me, let's say it's 65 or something like that. So if you have onset of depression in 20, 30, 40-year-olds, you don't have a big driver for later dementia. That could have many explanations. There could be no relationship. It could be a survival effect. But it doesn't seem that these earlier onset, largely heritable depressive disorders are really what we're talking about. In the Baltimore epidemiologic catchment area, and I think it's the only study that was able to look at this so far that I've seen published, there are two peaks for onset of depressive syndrome of major depressive episode. There's a young onset peak. There's a later peak after 50. I think it's that new onset 50s spreads out into the young 60s. That is a new entity that is arising in some people in the population. I think that new entity probably is the prodrome or the precursor or what occurs before the MCI or dementia that we see later. It's heterogeneous. I think the aging of the monoamine systems is probably very important. I think the occurrence of vascular brain injury, even at minor levels, in clinical states, we might see white matter change. That's also related to that. So that's why I'm saying we need to hone more to focus on, oh, and by the way, Panos didn't mention it, but there's a clear difference between the phenotype of depression you see in Alzheimer's and the phenotype you see in sort of garden variety late life depression. There's a wonderful publication out of Pittsburgh, probably now 2 decades ago. People who have depression in Alzheimer's dementia, they generally do not have suicidal thoughts. About a third of them are also delusional. They don't have much self-deprecating, guilt-ridden thoughts. Their peers who are the same age, who don't have dementia, who have earlier onset depression, have more of a more of a self-blaming suicidal phenotype. So I think that matters as well. So I'm purposely murking the waters because we've got to disentangle the age of onset, the phenotype, the underlying mechanisms that are candidates. And so we start to make our figures even more complicated. We have the tools at this point. We're starting to have the imaging tools. You've already pointed out about EVs as possibly very important tools. Vasso's work in trying to characterize in blood EVs of a particular cell origin. Can we see the EVs of serotonergic cells in the blood and see what's going on in those cells? Those are the kinds of tools that over the next 5 to 10 years, I think, will help us get more clarity on mechanisms.
Yes, I think we have booked with Vasso a talk on the HIAAD seminar calendar that will give us exactly this new triangle, trying to see individual cell with her work and the work developed in the field more, because that's also very important. Individual cells can produce different marks and footprints. I see, for example, from Dimitris Kapoyannis' story that says that in this particular disease state that has to do with dementia or cognitive decline, we have an astrocytic footprint on EVs and exosomes that can help us to the diagnosis or can relate it with the cognitive or any other behavioral problems. Thanks, Costa.
Other questions? There are no other questions. Thank you all. Thanks to the great speakers for the excellent bridging seminar. Thanks for the great discussion. Thank you all for being here today.
And we have to say, Savas, that the next seminar will be on 4th of May. So every first Thursday of the month, we are here. We've presented new, let's say, issues or triangles of this complex Alzheimer's disease pathology. And the next seminar is on 4th of May, Thursday at 5 o'clock Greek time, where Kostas Palikaras from the medical school and Evandos from Oslo University Medical School, they're going to speak about aging, mitochondria, and new generation, trying to go from a very basic to translational research related with new drugs and new mechanisms of aging, mitochondria, and new generation, all to the context of Alzheimer's disease.
Thank you, Yanni, for reminding this. Thank you all. Have a nice evening.
Thank you. Goodbye.
Noon in the States. Bye.
Noon in the States. Goodbye.
Bye.
Bye.
Bye.